In this video, we'll be looking at how to carry out the food test required practical. So we're going to be testing for sugars, lipids, proteins, and also starch. And the way that we do that is by using the different reagents that you can see all along the side here. We're going to start off with iodine. So iodine is used to test for starch. So within this tile, I've mushed up different bits of food. So in the first one, I've got some Rice Krispies. Then I have got some rice powder, milk powder even. Then I've got some custard creams mushed up over here, and then I've got some slightly rotten apples. So what's going to happen is we're going to add some of this iodine solution to each well, and we're going to observe what we see, so what kind of reaction happens. So I only need like a couple of drops, so I'll start with my Rice Krispies. What we can see here is that the Rice Krispies and the custard cream, they've both turned a sort of bluish black colour. Meanwhile, our milk powder and our apples, we haven't really seen a change because iodine was yellowish orange to start with, and that colour has not changed at all. So we've kept those two the same. So when nothing happens, we call that a negative result. The positive result is the change that we can see. And if our substance does change colour to blue or black, that indicates that we do have some starch present in the food or whatever we're using. So the next step is going to be to test for proteins. And this time we're going to use Biuret, so Biuret reagent. And we're just going to add a couple of drops again to each food sample. So what we'll do is start with our milk powder and pipette in just a few drops, maybe a little bit more just because it's not too clear. Sometimes it is easier to put a sort of white paper underneath, and then you can see the colour change a bit more accurately. So as you can see, what's happened here is that solution has changed to a purple colour. Now that purple colour indicates that we've got a positive test. So if we had no colour change, that would have been a negative test, but because we are able to see the purple, that is a positive test. So put that one away. Let's do our apples next. I think we do need a bit more than a couple of drops. So just put it underneath there. And as you can see, we can't really observe anything. There's no real colour change, so therefore that would be a negative test. Let's do Rice Krispies next. So we've got a cereal here, and we're trying to find out whether or not it's got any proteins in it. I can see something happening here, but I don't think it's a colour change to purple. I think what that Biuret solution or reagent has done is just kind of stained the Rice Krispies, it's not actually changed it to purple. So therefore we would stick to calling that a negative test. We have finally got our custard creams, so let's add some Biuret to them. Just swirl it a little bit. Maybe add a couple more drops. Let's have a look what's happened here. I think there we would say no real colour change. So the only one that we did observe proteins in would be our milk powder, because that's the only one that we saw a purple colour change in. So that would be our positive test. Next, we're going to be testing for lipids. So we've got lots of little test tubes and we've got some bungs at the top. You take the bung out and add a couple of drops of ethanol. Replace the bung and then just invert a couple of times and see what happens. Could possibly do with a bit more ethanol. And then again, just invert. So what we were looking for here is a sort of cloudy mixture, and that's generally what you see when fats or lipids are present. With this, it is hard to tell because we've got quite a lot of food products in there. So let's see what happens with the other ones first before we make a decision on that. Add a bit of ethanol here. Just invert. So with this, I would say the solution is clear. It's hard to tell just because the milk powder does make it look like it's cloudy, but if you look at the solution at the top, not sure if you can see in the video, but it is actually clear. So that would be our negative test. Let's do Rice Krispies. See what's happened here. Now again, in the Rice Krispies, I would probably say that that's still a clear solution because at the top the liquid that we can see is still clear. Yep, so that'll be a negative test. Finally, let's have a go with our apples. Sometimes you can actually predict what's going to happen just because certain foods, you know, will contain a lot of fats or a lot of sugars or a lot of proteins. So you can often predict the results. Yeah, as expected, our apples are non-fatty, so they don't have that cloudy kind of liquid. So therefore that's a negative test. Going back to our custard creams, that solution does look a lot more cloudy now, so we can refer to that as a positive test. And that will mean we have got some fats in our biscuits. Finally, we're going to be testing for the presence of sugars or for glucose. So I'm going to get my boiling tubes and add our reagent, which in this case is going to be some Benedicts. So I'm going to add a couple of drops of Benedicts. And then to add some boiling or warm water, hot water even, to my boiling tube. And usually, if there are sugars present, the solution turns orangey red after a while. So we'll just leave that and see what happens. So going back to the summary, we've looked at iodine. So iodine is a reagent for starch presence. So if starch is present, we'll see a color change of blue-black. Next, we have got our biuret. So biuret is for proteins. So if we do have proteins in the food sample, then you should normally see a color change to a pale purplish color. Then we've got some ethanol. Ethanol is the the reagent for testing lipids or fats, and you would expect to see a milky kind of solution by the end. And finally, we have got our Benedict. So when we add this in combination with hot water, we see a color change, well, we should have seen a color change to a sort of orangey red color. And that's it for this video.